So, yesterday we began starting to talk about some of the biochemical compounds, in this case sugars. Okay, so one thing that I want you to maybe make note of is when we're looking at these structures, there's a simple way, well, I think it's simple. There's different types of ways that you can say things to help you remember them. So that would be for yourself, 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 and yourself. Okay? So of you four, who do we pick on? Let's ask everyone else. Of those four, who do we pick on? Well, I know one of them. I'm asking you, which specific one? Okay. How are proteins digested in the small intestine? Okay. Pepsinogen is a clue. Okay. Hydrochloric acid is a clue because they come from specific types of cells. Parietal's one. Chief is the other. So now that you got the two cells down, one of them secretes pepsinogen, the other one secretes acid. Little help? Why is parietal acid? Because there's an A in parietal. There isn't an A in chief, okay? So then the chief cells secrete pepsinogen, which then it's activated by the acid. So it's things like that that might help you out, just like in this case. When I think of this, whether you say research and development or Reader's Digest, hopefully that can be a simple analogy when it comes to drawing these structures. Does this look like anything that you've seen? No, it's not. Because the ones that you have seen are this one. I'm not going to put the hydrogens in there, okay? So then I bet this one looks familiar. And that's glucose, okay? Then just by switching a couple of these around, okay? Another G, that's galactose, okay? So then if we go back to our first form that we had, back to glucose, now that we've had time to visualize this, so here's carbon number 6, okay? And here's carbon number 1. So yesterday we did alpha D glucose. That means this has to be down, okay? It's because if this is flipped, okay, that's a specific term we call maturation, which means then you can bond that to, let's say, another glucose monomer, and that's how cellulose is made, okay? Because it matters a lot how this carbon atom is oriented on here, because it totally changes what it can bond to, bond with, how it bonds together. But when we look at this, carbon number 2, okay, so we apply this, okay, so since it's on the right side, where is that OH molecule, up or down? Right, okay, then we look at carbon number 3, which is right here, okay, and then here's carbon number 4. And then there's a couple of different changes that went through, and that's actually a hydrogen atom there. So that's how you would draw alpha D glucose. All right. So let's use that same knowledge then and do that for this molecule, which is, as we said, hang on here. Okay, so then we can do this for this mannose monomer. Okay, so do we want this to be an alpha or a beta monomer here? We've seen alpha. Let's make that beta then. So that means this is on the top side now. Okay, so let's look at carbon number 2, which is right here. Where's that OH molecule? It's on the top or top side. Okay, carbon number 3, which is right here. Top side or bottom? Okay. Sometimes forget to put those hydrogens in there. Okay, here's carbon number 4, which is right here. Yeah, because it's on the right side. Okay, and then there's a couple switcheroos that took place, and that hydrogen atom ends up down there. Okay, so ones that you need to know how to draw from memory would be glucose, galactose, and don't be too concerned about mannose. You'd have to be given this orientation here, okay? This one is called mannose. Okay? In this case, you would do that, yes. Beta D mannose. I think there's 2 N's in there. That's actually a good question, so let me quickly take a look. Yes, 2 N's, M-A-N-N-O-S-E. Okay. And we're not going to do a whole lot today. We're just doing a slight review. So any confusion with anything that is to the left of this? Okay. Front row, are we good? Gesundheit. Second row. Third row. Third row. All right. So this next one is probably, if not one of the most important sugars, maybe next to that of glucose. This one's a little more tricky though. With our limited knowledge so far, I should have put this up here. Are these aldose sugars or ketose sugars? That's how we ended yesterday's presentation. Yes, these are aldose sugars because this double bonded oxygen is on the first carbon, just like any other aldehyde that we've ever looked at before, just identifying the organic family. Okay, so how many total carbons are in this atom or this molecule? There's 1. Every intersection there. It's kind of a trick question. We haven't spent a lot of time on this, so I can see why this would be a little confusing. So we've got 5, okay? So if these have 6 carbons, they're a 6-sided figure. If this has 5 carbons, it must be a what-sided figure? Yep. Or a what? Pentagon. Okay. Okay. And that's right. I'm just going to try to clean this up just a little bit. Try to make it maybe more so equilateral, if that's possible. I think that's a little better. Okay, so oddly enough, there should be an oxygen up here. Okay, so we're going to use the same knowledge that we've done. We'll just make this a beta molecule, or excuse me, an alpha molecule. Okay, so there's carbon number 1. We're going to put it on the bottom. Okay, carbon number 2, which is right here, does that go on top or on the bottom? It's on the right side, so it goes down. Okay. Here's carbon number 3. It's on the right side, so it goes on the bottom. And with practice, we'll get really better at recognizing this because this oxygen is that one right there, but we still have to account for this, so that's actually up top there. Because these had gone through a process, we were really specific with that yesterday. Okay. Therefore, this molecule is that sugar. Okay? And if we do this, if we take that oxygen off of there, now it's a really, really important sugar. Because now this is lacking in oxygen, and it's at the second carbon, so we would call it this. Because we took that off, then we put this term together, and then if this is bonded to a phosphate bond and other nucleotides, what's this possibly referring to? Get rid of this. Holy cow, it's a long word, right? So it's the sugar that's found in this, and that. So why are we underlining them? This is the main sugar found in DNA. That's why we said very important molecule. Not just because of DNA, but also what other nucleotide strand? RNA, Gesundheit. Okay? So from a biochemistry standpoint, some of the terminology that we used last year in biology when it came to protein synthesis now actually sort of comes around full circle to where this is one of the important molecules needed for DNA. Tough stuff. Right now, I could see this would be a little difficult, a little intimidating, just because we really haven't spent a whole lot of time on that.
Okay, on that last one, you know, when you have CH2OH,
Mm-hmm.
like always on your last carbon, is it always that CH2OH and then an H on the bottom?
Yes,
Okay.
that's correct. Okay, we don't need to. We showed you that there's like 3 steps. The more I think about this, okay, with practice, we'll begin to be able to recognize what happens here, what happens here, and then what happens on this one. These dashed circles that we're circling, on these structures here, what do they all have in common? Not the number. So, repeat that. Yeah, well, so are these here. It's the last one. Okay? It's the bottom one here, and bottom one here. Okay? But we'll make sense of that a little further down the road. I think with practice you'll be able to recognize this. It's just for the biochemistry class, we want you to be able to understand that intermediate step. We're not illustrating that here. And I think for our purposes, I think that's better for you as well. Because this isn't, it's just a short, you want to call it biomolecule section, rather than just spending a lot of time saying this is the biochemistry behind it. That might be a little more intimidating. Okay, we did not draw sucrose yet, but I think it would be better off. We could do that today. I mean, yes, we could. But I think it would be better for once everyone gets back here, which tomorrow at this time, let's see here. We've got 1, 2, 3, 4. Then it would be 5, 6, 7, 8. 8 of you are gone, correct? Did I miss anybody?
What time do we eat?
So 9 then. Okay. Yes, we will be loading the bus probably about 1:10. Be in here for a little bit. And then hop on the bus, drive on up there. That will be quite an experience that you will be able to take part in. I don't think Madison got to do that. Maybe we went. I don't know. But for those tuning in later, go to the anatomy lab up at SDSU. So that's what we'll be looking at. Okay. Later on this week, we'll perhaps start talking about some of the amino acids. We'll wrap this chapter up next week, and then it's time to start reviewing for finals. Okay. Any questions on this? All right. So that's all I have. We'll catch up to you next time.